Hello, everybody. My name is Marie, and my presentation today is going to be on the disease hepatitis, which is a disease of the liver. There are a few causes of hepatitis, including chronic use of alcohol, toxins, and chemicals, and there is an autoimmune condition that can cause hepatitis. But in this presentation, I will concentrate on the viruses that cause hepatitis. And I hope that you will learn the various types of hepatitis, the structure of the hepatitis virus, the mode of transmission, current research connecting the microbiota with hepatitis, and the history and health concerns. And here we have a picture of the liver, and you can see in each of these diagrams and images, there is a healthy liver and one that is damaged by hepatitis. And you can see that the lobules and the scar tissue that results from any kind of infection with the virus, any kinds of the hepatitis viruses. And your liver is a really important organ. The word hepatitis itself means inflammation of the liver, so it can be from anything. Our liver is a very important organ because it detoxifies metabolites, synthesizes proteins, produces biochemicals for digestion, regulates glycogen storage, decomposes red blood cells, produces hormones. So it does a whole host of functions, and it is the largest organ in the body along with the skin. So this is what happens when one gets damaged by hepatitis. This is a chart showing the overall view of the 5 different kinds of hepatitis, although they do think that there is even more than A, B, C, D, and E. And this is a nice overview because it shows here the family names and what kind of genetic material it has. Also shows the route of transmission and the general age of people who are affected by each of these types of viruses. And what I would like to do is go through each of these types in a little bit more detail. Hepatitis A is caused by this hepatitis A virus. It is single-stranded RNA and lacks an envelope. It multiplies in the epithelial lining of the intestinal tract, and it can spread to other organs. The virus is shed in the feces, so it is called the fecal-oral route of infection, and one common route is with food handlers. Everyday concentrations of chlorine do not kill this virus. Symptoms include loss of appetite, malaise, nausea, diarrhea, fever, chills, and abdominal discomfort, and can last 2 to 21 days. The liver becomes tender and enlarged, and development of symptomatic hepatitis A is correlated with patient age. Few children under the age of 6 actually show symptoms, and the majority of adults will show symptoms that persist for 2 to 8 weeks. Hepatitis is seen more in lower socioeconomic populations. It can be detected by observing immunoglobulins M and G, anti-HAV antibodies, which appear about 4 weeks after infection. One interesting note about this virus is that it has a quasi-enveloped form of HAV, which is detected in the serum and plasma of infected hosts. But a non-enveloped form, a naked form, is actually shed through the feces. So apparently the virus loses its envelope following exposure to high concentrations of bile salts, which are produced in the liver, in the biliary canaliculus, which is a part of the liver bile duct. It's a channel of capillary tubules that form the bile duct. And in this way, the HAV evades the immune system. And then this semi-envelope sequesters the virus from the neutralizing antibodies that target capsid proteins. The viruses that are shed are stable and highly transmissible. People can acquire a lifelong immunity to hepatitis A. HAV vaccination is now part of the childhood vaccination schedule. Hepatitis B is caused by the HBV virus. Unlike hepatitis A, the virus is caused by hepatitis is larger. It has a double-strand genome of DNA, and it goes through an intermediate RNA stage that resembles a retrovirus. Scientists have studied HBV because it has been transmitted in blood transfusions, any blood fluids, sexual contact, and contaminated syringes. It can be fatal and is difficult to diagnose because the symptoms are much like the flu. You get mild fever, headaches, fatigue, abdominal cramps, and loss of appetite. It is considered a major health problem because over 400 million people have chronic hepatitis B. And you can see these particles over here on the right, which I will talk about in the life cycle right now. I chose hepatitis B to show you how a typical hepatitis virus gets into a cell, hijacks the cell, and this is a little bit of a complicated one here because it produces different kinds of this circular DNA. If we start up here on the right side, the HBV virion over here, we're going to move from the right to the left, binds to the surface of the liver cells or hepatocytes at the cell-associated heparin sulfate proteoglycans, pre-S1. Endocytosis occurs, or fusion, into the cell, uncoating the nucleocapsid there, and then the relaxed circular DNA is repaired by both the viral and cellular enzymes. And then this incomplete DNA strand is completed by the viral polymerase. Then the viral polymerase and RNA primers used for DNA plus the strand synthesis are removed by cellular enzymes. Covalently closed circular DNA, which is this CCC DNA, is formed by covalent ligation of both DNA strands. There is evidence that infected cells contain between 1 and 50 CCC strands of DNA that exist as little mini chromosomes. And then the CCC DNA now serves as a template for the RNA synthesis. There are two phases of hepatitis B, acute and chronic. Acute hepatitis B is when a person notices the symptoms within 1 to 4 months of acquiring the virus, and it may resolve itself on its own. 9 out of 10 people recover from infection from hepatitis B. Chronic hepatitis B lasts longer than 6 months, and a blood test can show a definitive prognosis. The serum of a patient with HBV has the 3 particles, which I showed you in the last diagram, and that's the Dane particle, which is the complete virion, spherical particles, and filamentous particles. These partial particles still have the antigens, which can be detected with the antibodies. A vaccine is available for hepatitis B, and it is given in a series of 3 shots starting in childhood. And although we have this vaccine, about 630,000 people worldwide die from hepatitis B. Hepatitis C is an enveloped single-stranded RNA virus that either causes an immune response and clears the infection or slowly destroys the liver. It is known as a silent killer because so many people go undiagnosed and do not realize they have it until about 20 years after infection. It kills more people than AIDS in the United States. One problem is that there is a delay of 70 to 80 days between infection and the ability to detect antibodies. Infection with hepatitis C is the #1 reason for liver transplantation. According to the NIH, several newly developed drugs can cure about 90% or 95% of all people infected with hepatitis C. There is not a vaccine to prevent infection from hepatitis C yet, but it is coming soon. Hepatitis D is caused by viruses in the Delta Viridae family. It is a very unique virus that is defective and can only inflict harm on hepatocytes if it is used in conjunction with hepatitis B to complete its life cycle. It is an RNA virus that does not encode its own protein envelope and uses the hepatitis B virus surface antigens to do so. And you can see both the B and the D in this diagram. It can infect a cell and can replicate itself, but cannot secrete infectious particles. Interestingly, HBV vaccination works for HDV, also. Several antivirals targeting other parts of the life cycle, for instance, and enter into the hepatocytes. Hepatitis E is caused by the pathogen Caliciviridae, and it is the most common form of acute hepatitis. Chronic conditions occur in immunocompromised individuals and in patients with solid organ transplants. Ingestion from food or water contaminated with fecal material occurs, and we find this rarely in the United States, but in poor developing countries, mostly in South and Central Asia, Sub-Saharan Africa, and the Middle East. Most people recover from hepatitis E, but oddly enough, in pregnant women, if they have the virus in their second and or third trimesters, they have a higher risk of death or miscarriage. In fact, in excess of 20% of pregnant women with HEV will die, and a vaccine for hepatitis E has been developed in China. I would like to share a study connecting the gut microbiota and viral hepatitis, cirrhosis of the liver. And it seems that microorganisms in our GI tract and really all over our bodies have play a role in our health, as we've seen in the news quite a lot lately. And this review study of about 50 experiments looked at the relationship between the gut biome and cirrhosis caused by hepatitis. Emerging data suggests a significant role of gut dysbiosis in viral hepatitis cirrhotic patients with a decreased form of certain kinds of microorganisms, specifically Firmicutes, and an increased population of bacteroides, populations of bacteria. Research have a gut dysbiosis index as a way to measure populations of microorganisms in our guts. And as we've learned in this class, the human gut has trillions of bacteria, fungi, archaea, and viruses that are busy doing quorum sensing, retaining water, horizontal gene transfer. And some researchers are trying to use this microbiota index for diagnosis and prognosis in cirrhotic patients. This diagram here shows a healthy and cirrhotic liver and what they think is happening in each. Products from microbial metabolism influence and modulate the host immune system through two pathways, and they are the PAMP, which is the pathogen-associated molecular pattern, and the DAMP, which is the damage-associated molecular pattern. And what is happening is the gut barrier, which is shown down here, is showing that it's altered, and substances are then able to move through the portal system, having a deleterious effect on the liver. So, difference between the healthy gut lining and the unhealthy one. Hepatic encephalopathy is a serious complication in viral hepatitis cirrhosis, and it has been found that these patients have an increased Haemophilus, Veillonella, Clostridium, and Bacteroides, and reduced forms of populations of Fusobacterium, Magamonas, and Fecal Bacterium. So there is a unique gut microbiota of viral hepatitis cirrhotics. FMT, which is fecal microbiota transplantation, probiotics, prebiotics, can restore microbial diversity in hepatitis patients. And here are a little bit of history and some health concerns. People have varied reasons as to why they get vaccinations, and there was one study by Casting et al. Who studied whether healthcare providers gave a gain-framed or loss-of-framed message to patients. And the difference between these two types of messages is how the healthcare provider frames their message. And we all know when we go to the doctor what we are like from a patient's perspective. And they wanted to know, is there a clear positive advantage to getting the vaccine, or should the healthcare provider talk about the negative effects of not getting the vaccine? And the study found that a strong provider recommendation, as opposed to an offer and just suggesting that you get this vaccine, was better advice for patients. There are diagnostic tests for hepatitis, most of which involve detecting immunoglobulin antibodies. And the various types of hepatitis were only really known after World War II. The HBV vaccine is the first vaccine not made from tissue culture, but from plasma. The HCV virus was discovered by a direct molecular approach without tissue culture, electron microscopy, or serology. In the 1960s, Baruch Blumberg discovered hepatitis B, and the HBV vaccine was the first vaccine that was an anti-cancer one. And I am very excited to tell you that I just realized, with the Nobel Prizes given out just this past 2 weeks, that 3 people, Harvey J. Alter, Michael Houghton, and Charles M. Rice, recently just won the Nobel Prize in Physiology or Medicine, who discovered the RNA flavivirus hepatitis that causes hepatitis C. And you can read more about their discovery on the Nobel.org website, which is a very, very interesting website. Highly sensitive blood tests and rapid development of antiviral drugs are now available because of their work and research. So I hope you learned more than you knew already about hepatitis. And here are my references. Thank you.